for our discussion on phenols.
Okay, thanks, Cindy. Welcome, everybody. I appreciate you taking the time out to attend and listen to this. Let me just preface this talk by saying that this has been a real eye-opener for me. And the material is very complex, even for a Ph.D. level biochemist. And I feel like that I may be a little bit over my head on the understanding of this. So let me give a little background. When we came out with our No-Phenol enzyme product, a lot of parents noted that their children were able to tolerate fruits and vegetables and even natural food colorings and even artificial food colorings and those type of things better than previously without the enzyme. And this has always been in the back of my mind to kind of investigate this further, and so the last couple of weeks I've been trying to do that. And what I do is I basically do literature search. I look through the medical literature just on PubMed and other sources and see what the existing literature has to say. And a lot of this I do, I tie together different articles, interpret data from different labs and research areas and try to develop a working hypothesis. So a lot of the things we're going to talk about is not to justify a product or to sell a product. It's to give you information to help you understand what's happening with you or your child's digestive process and why you may be seeing certain problems. Whether an enzyme product or any other product out on the market helps, I can't give you an absolute guarantee or answer, but I'm hoping that this brief touch on this subject will just kind of point you in the way to start looking at this problem. So what we're going to do is, again, this may be a little bit confusing and I'm not sure I understand it fully, but again, I don't want to feel like I have no confidence in this, but I'm just amazed at the complexity of the thing. But anyway, it'll just help you understand, I hope, a little bit about this. And I think in the future on our newsletter and blogs and website, I'll be trying to address this further as I develop these ideas and problems a little bit better. Okay, having said all that, first of all, make sure that I've now switched to slide number 2. So I hope you're all seeing that. It's titled Level of Enzyme Supplementation Help. If you can't see that, please type in the chat area and let us know. The title may be somewhat misleading. I hope I didn't lead anyone astray thinking that we have the answers through enzymes on a lot of these problems. I think there is some indications that in some instances enzymes can help. But again, I just want to throw some light on the whole issue and maybe kind of separate out some of the different areas and show where enzymes or other things like probiotics may be helpful. So we're kind of concentrating on 3 areas: lectins, which are sugar-binding compounds, oxalates, which are basically derivatives of oxalic acid. If you have trouble with kidney stones, a lot of it could be calcium oxalates. It's a byproduct of metabolism, and the body should have a way of degrading and removing it, but sometimes that goes awry. We're also going to be talking about polyphenolic compounds, and I think this is the area we're going to be concentrating on the most today. And we also want to differentiate between polyphenolics and phenol. They're very different. Polyphenolics are good, they're antioxidants. Phenol is not, it's a toxin, and so we're going to be addressing those a little bit. As far as enzyme supplementation, I've sort of put this here. Lectins, we don't know. Right now it looks like help as far as oral enzymes, probably low level. But again, that's sort of a gray area. Oxalates, enzymes could be helpful, but I think it's the enzymes that are derived from certain strains of probiotic bacteria that are found in the gut. And this has been addressed by people like Susan Owens and other people who have developed a low oxalate diet. So I think this is an area where probiotic enzymes, or if we can find the enzyme that the bacteria produce, if we can find that enzyme in fungal enzyme blends, then it could possibly be that oral enzymes could be used in this. But again, we haven't verified that. And the polyphenolics, this to me is quite interesting because it overlaps into the area of the sulfation and methylation area. So the phenyl sulfosyltransferase enzymes and that type of thing. Also the integrity of the gut mucosa, interestingly enough, comes into play because, as we'll see, the mucosa is the area where a lot of the sulfation enzymes reside, and that's where a lot of the conversion of these polyphenolic compounds to their active form occurs. So we're going to show how just generalized gut health and why a damaged gut wall may be the reason you see a lot of these problems with fruits and vegetables. Okay, lectins are very interesting compounds. The term lectin comes from the Latin word legere, which means to select. And that's what lectins do. They select out certain compounds. And they select based on the type of sugar groups found on glycoproteins and glycolipids. So lectins are also the way we determine our blood group, A, B, and O. Type A has different sugar groups, type B has another type, and type O has different ones. And even when you get down to O negative and positive, those are different sugar groups on the outside part of the red blood cell. And those sugar groups are attractive to lectins. So lectins can be used to select out different cells. In fact, in the laboratory, we call it a con A column. It's basically a lectin called concanavalin A, which, when bound to a solid matrix, we can use to purify glycoproteins. So based on the different sugar group that we find on the lectin, we can purify or select out different glycoproteins. Because of their ability to select different compounds, the lectins are very much involved in biological recognition of different cells and proteins. And again, the lectins are extremely large, very diverse group. They provide both nutritive and anti-nutritive properties, and we don't want to paint lectins as something bad and evil. They do have a role, but as in a lot of things, an excess can cause problems. What lectins do, they have a variety of functions. One, they regulate how cells stick to things, called cell adhesion. They're also involved in which glycoproteins are formed. They do bind the soluble glycoproteins, those in solution, and glycoproteins that may be found on the outside of cells, like those on the outside of the gut epithelial cells. And so this is where we can get some problems, because some of these lectins, if they bind to certain gut cells, they can cause different things to happen. In some cases, so what happens is not bad, but in many cases it can, and also determines, and what it does may depend on just the general condition of the gut as well. They target proteins for delivery to lysosomes. Lysosomes are cells that are basically the garbage men of the body. They go around and scavenge up damaged cells, damaged proteins, bacteria and stuff, and then they have proteases inside the lysosomes that basically are recyclers. They just tear it up, recycle what they can, and throw out the waste. So they are important. The lectins can grab a protein, a damaged protein or cell, and then basically whistle the lysosome to come over and pick it up. So it kind of helps the process, basically. Also, pathogens, the virus and bacteria often have, especially viruses, will have specific glycoproteins on the outside of their cells, and or they have specific carbohydrates, and the lectins in the body can bind to these carbohydrates, and so then that interaction between the lectin and the pathogen cause other functions to occur, such as IgE and IgG reactions. It can also stimulate a Th2 allergic response as well. And we'll get into this a little later, but sometimes the lectins can be involved in some of the leaky gut reactions. So they are involved in different ways into allergy reactions that aren't picked up by traditional skin prick tests or allergy testing. And the problem with lectins is that many survive the digestion. The enzymes that the pancreas puts out and such really aren't designed to break down lectins. And oftentimes they survive simply by binding to the gut cell, and that kind of protects them from being degraded. And again, we want to emphasize that we don't want to paint all lectins as being bad or harmful. So we shouldn't fear them. And a lot of the toxic effects of lectins are due to basically overconsumption. In fact, there was an interesting article in the British Medical Journal back in 1999 by an allergist named David Fried, who mentioned that there was, in 1988, a hospital launched a healthy eating day at their cafeteria. And one dish they served contained red kidney beans, and 31 portions were served. Over the next 4 hours, a total of 12 people suffered profuse vomiting, some with diarrhea. All had recovered by next day. No pathogens were isolated from the blood or isolated from the food, so it wasn't food poisoning. But the beans they used contained a abnormally high concentration of the lectin PHA, or phytohemagglutinin. And so again, this is something that probably bothers a lot of people without them knowing. Eating a meal and then getting sick, they just may attribute to food poisoning or E. coli or something like that, when it just could be a reaction to the lectins. Slide 5 here shows a sort of a cartoon of how the lectins bind to the gut cells. We can show that these figures here are the cells of the gut epithelial, and these little purple balls here are basically the lectins, or particles that have lectins on the outside of them. And they interact with the sugars that are found on the cell outside of these cells. And so this is how it would appear if you magnified that. This would be the sugar on the outside of the cell, and that's the lectin interacting. And when that happens, that interaction causes things to happen down inside the cell. Okay, the toxic food lectins, such as beans and grains, seeds and nuts, can be harmful if consumed in excess. Again, small amounts are probably not going to cause a lot of problems in the general population. And often, not all, but just cooking food can inactivate a lot of harmful lectins. And this is something you have to be aware of if you are one of those people that are really into raw foods, which it sounds like a good idea, but if your gut is damaged or if you are sensitive to these lectins, you're going to get a higher dose of them. So that's why I think a lot of people have problems eating raw foods, especially raw veggies and such, raw beans or stuff. But if they have it cooked, they're fine. Again, a lot of these lectins are involved in allergic reactions, nutritional deficiencies, and the immune reactions that we mentioned earlier. And again, the effects may be dependent on the blood type, and that's why you've seen the books called Eating for Your Blood Type and those type of things, basically spouting the hypothesis that if you're type A or B or O or whatever, you should avoid certain foods. And the idea is those foods with certain lectins that bind to your blood type may cause problems. Now, I'm not sure I buy into that fully, but it's an interesting hypothesis, and it's something that certainly someone can try. A lot of people on these type of diets espouse that they feel much better and don't have the problems that they did. So there may be something to that. Another idea is that you can block these lectins, since these lectins do bind or sugar binding compounds. If you take along some of the sugars that, like mannose, that would bind to the mannose binding lectin, you can basically keep them from binding cells. So another way to think of this, and this is an area where I believe enzymes can help. If you're taking enzymes that break down starches, which produce oligosaccharides and sugars, then you're probably going to increase the likelihood of those lectins in the food being bound to those sugars found with the food. So that may be one way that some of the high carbohydrates enzyme products are helping. Let's see. I'm going to take a break here. Cindy, am I doing okay? Are we having any problems?
You're doing fine.
Okay.
Are you able to see the questions?
I cannot see any questions.
Still cannot. Okay.
No. So just keep track of them, and we'll get to them as soon as we get to the end here.
Okay. We'll let you take
Okay.
I didn't know if you needed to take a quick break, or are you fine?
No,
okay,
we're fine. I just want to make sure that that I was not talking into empty air.
Nope, doing great.
Okay. Again, health implications on slide 7 here. I did preview some of the questions. There was an interesting question. Someone wanted to know if the lectin issue overlapped into the area of gluten, and I'd say yes, because gliadin, which is a peptide derived from gluten, and this is the peptide that presents problems with celiac disease, does have lectin properties. It binds to the intestinal mucosa. So in addition to its other properties, it can also be binding to the sugars on the cells in the intestinal tract. There are some grains such as wheat and soy tend to have diabetogenic properties, and there are several papers out there that are hypothesizing that lectins may be binding and having effects on if they get out of the gut. If there's a leaky gut problem, they get into the systemic circulation. They can bind to certain organs like the pancreas and affect the insulin production. So they're and in fact, my wife, who has recently diagnosed with type 2 diabetes, her endocrinologist basically asked her to cut down on wheat and gluten products. And that may be one of the reasons why you're trying to protect the pancreas as much as possible. And here's another area that I think a lot of parents have reported to us that enzymes help their children who have the red cheeks and red ears. That seem to occur with certain fruits and vegetables. And the red cheeks and red ears, the only thing I can think of is that the only thing that really causes that is a release of histamine that opens up the small capillaries on the face and head. So, and that's why you see you just get increased blood flow. That's why the ears feel warm and turn red. Lectins can be the cause of histamine release. They can bind to certain mast cells and other immune type cells and cause a massive release of histamine. So that may be why that's occurring. Also, this is, I think, very important for those kids on the spectrum who are having GI problems, that the lectins can cause damage to the inside of the gut, basically removing or debriding the mucus layer on the gut. And the more I read on that, uh, I am impressed with the importance of the mucus layer in the gut. It's not just a slimy layer that protects from acids or whatever. It also, there's a lot of stuff that goes on in the mucus, and we'll get into more of that. But this is a very protective layer. I think it's sort of a suicide trap. It will sacrifice itself to protect the tissue underneath. So I think lectins can bind to sugar groups in the mucus, and that's better than the lectins binding to the cell itself. So I do have a little bit of concern over some products that talk about removing biofilm, because biofilm is basically has the same consistency or same composition as mucus. So we want to make sure that any product that addresses biofilm can basically tell the difference between normal mucus and something that's biofilm in nature, because we do want to protect that mucus layer. Also, interesting enough, this is another area of concern. If we were talking about genetic engineering of some of our foods, it's something I'm not aware of, but prohevin, which is a lectin and also the main allergen of latex, has been engineered into transgenic tomatoes because it's an excellent fungicide, or it keeps fungal growth to a minimum, and that may be fine for getting a very nice prize-winning tomato. But I think if we're basically producing tomatoes that have a potential allergen, and if you think of the amount of latex allergy in the population, I think this could be a matter of concern. So that's one reason why we're a little reluctant to recommend genetically modified foods in this industry. As far as the lectins and enzymes, there's some other reasons that certain foods might be well to be avoided or need extra oral enzymes to compensate. One would be that all legumes, beans, whether they're lima beans, fennel beans, brown beans, or whatever, they contain levels of an enzyme inhibitor, amylase inhibitor. Amylase is the enzyme that helps to break down starch into simple sugars and oligosaccharides. And there is a very potent lectin called phytohemagglutinin. It's one we mentioned earlier. It's also an inhibitor, binds to amylase and makes it inactive. So that could be one reason why a lot of people have problems with legumes, and they get gastric distress when they eat high amounts of them. Another interesting thing I ran across was that lectins seem to have influence over gut development and immune function. And animal studies seem to show that given, I think in certain mice, very young baby mice, found when they were given a very high diet high in lectins, they saw the gut mature much quicker, which may sound like a good thing, but actually it may not be. And also there were some differences in the immune function of the gut as well. So very interesting. Again, it just shows that lectins can have a powerful influence over the gut. Also, the disaccharidase enzymes like lactase and galactosidase, those type of enzymes seem to be affected as well. And in some cases they were increased, but in most of them they were decreased. And again, it seems to be some variation species to species, as well as the effect of different lectins. But this may explain why so many people on the SCD diet and other low-carb diets seem to have such good results. So when they have a low-carb diet, they usually, the ratio of protein to carb is much higher. So protein is higher, and so we see less of an effect of any lectins in the diet. So that may be why the SCD can produce good results. And I also think those enzymes that break down carbs and such may again have some influence over the lectins as well. Very quickly, I'm probably spending more time on lectins than I wanted to. This is just a chart showing what lectins can do. They bind to gut cells, interact with enzymes. They can make changes in organs, changes in the mucus layer, and all these kind of go to different things: alter metabolism, poor protein digestibility, increased cell turnover because they're binding and damaging cells. And this can ultimately go to things like nutrient malabsorption, impaired immune function, and poor growth. Can enzyme supplements help? I know that's probably the big question. The fungal enzymes are slightly different in function from the human gut enzymes, which don't seem to do much good. So the addition of enzymes from another source, i.e. the fungal organisms, may be able to provide more of a digestive aid than the pancreatic and intestinal enzymes. And while the lectins seem to be resistant to proteolytic degradation, certain of the carbohydrates might be helpful in removing the sugar binding component of the lectin or affecting whatever the lectin binds to, or just increasing the amount of sugar or oligosaccharides, free saccharides that can bind to the lectins and basically keep them from binding to important parts of the gut. And that's what this says. Production of simple sugars and monosaccharides from starches by amylase may act to bind the lectins, keep them from binding to gut cells. For this, I would recommend cellulase and xylanase enzyme products, not because of anything these specific enzymes do, but these have what we call side activities or other undocumented enzymes that I have some idea of what they are, what these enzymes are. But these may be playing a role in affecting the lectin. But again, we haven't verified that. And just want to remember that, lectins can be toxic at high dose, but it's not something we need to fear. You can modify the dose and keep them to a minimum in diet. If you are sensitive, not everybody is sensitive to them. So very quickly, oxalates. Oxalic acid is a toxic compound produced in large quantities by cellular metabolism. It's also found in foods like spinach, Brussels sprouts, and such. If it accumulates, you can see some pathology, most likely things like calcium oxalate stones in the kidney. Again, my wife's side of the family has this problem quite a bit. So the thing is there a mechanism for removing oxalic acid? And yes, there is. While the humans don't produce any kind of enzyme or something to address oxalic acid, but the bacteria that is present in our gut, deep in the colon, do produce, or there's a species called Oxalobacter formigenes. If it is absent in the gut, you often see higher levels of kidney stone formation. So this bacteria actually produce enzymes that degrade oxalic acid. And those bacteria are usually the lactobacillus. Interestingly enough, the bifidobacteria don't appear to have the specific enzyme to degrade oxalic acid, but there are certain strains of lactobacillus that do it. Again, not all strains of lactobacillus, but there are some out there. The low oxalate diet can produce positive changes in behavior, espoused by many. Susan Owens has led the forefront of this. And again, the question, can enzyme supplements degrade oxalates? I don't know for sure, but here's what I do know. Aspergillus niger, or A. niger, is a major source of the enzymes. In fact, that's the major source of all commercially available enzymes. So a lot of A. niger is used to produce a lot of the different enzymes that we take in enzyme supplements. I found several papers stating that actually fungal organisms produce a lot of oxalic acid, but they have an enzyme for breaking down the oxalic acid. And that enzyme is oxalate decarboxylase. Converts oxalate to formate and carbon dioxide. And I should reassure you that in an enzyme supplement there would be no oxalic acid because of the way they purify the enzymes. Being a small molecular weight compound, oxalic acid is easily removed. But the organism does have to produce an enzyme to address that. Now here's my hypothesis, and this enzyme is active under acidic conditions. So if it does happen to be present in undocumented form in enzyme supplements, it could potentially be active in the gut. And we do need to verify this, but again, anecdotally, there are some people that report help in having lower oxalate levels with enzymes. Now, this is an area that I really find interesting and want to get into. Polyphenolic compounds. These are very, very abundant in the diet, and there are several hundred identified in foods, and there's probably several hundred more that we haven't found. The chief interest of researching these is because of their high antioxidant potential, and you see this all the time. You see cranberry commercials. You see a lot of articles written about ACAI. I'm not sure how you pronounce it. A C, I think. Yeah, a new berry. That's the berry du jour now, one that has high levels of antioxidants, and now you're seeing in all types of drinks and health bars and such. But the polyphenolics modulate activity of a wide range of enzymes, receptors, and other pathways. But mostly we're interested in because they can keep down the level of oxidative stress in mammals. So this is a very hot area of research right now. Now, the thing that can overwhelm you is looking at the differences. There are so many different types of polyphenolic compounds. There are phenolic acids. Here's a couple. Hydroxybenzoic acid, that's another group. In that group, there are gallic acid and protocatechuic acid. Another subtype would be the hydroxycinnamic acids. And these are I'm more familiar with these: caffeic acid, chlorogenic acid, ferulic acid, and chimeric acid. And these are what you're going to find mostly in most fruits and vegetables, very high in. Another type of polyphenol is flavonoids, and again, you have several different classes of flavonoids. Phenols. You probably may have heard of quercetin. I think you can buy that as a dietary supplement. Miracetin is another one. The flavones, apigenin, and luteolin, you find these in the bright yellow fruits and vegetables. Isoflavones, this is what is espoused for soy products. Diadzine and genistein are estrogenic. They have slight estrogenic activity, so a lot of women take this to help them with low estrogen levels. Flavonones, there are two that are interesting. Naringenin is found in grapefruit and citrus, as is hesperidin. I think that's also found in beets as well. Another type is anthocyanidins. And cyanidin and petunidin are levels. And the flavonols, which are the catechins and gallocatechins. And another group, still beans. You've probably heard of resveratrol, but this is the polyphenol found in wines and grapes that is thought to be why you can eat a lot of fat or be on the Mediterranean diet and that type of thing, and thought that this is the substance that protects from oxidation. Lignans, I'm not even going to try. That's a resinol type of antioxidant. I have no idea how to pronounce it. But these two are probably the least amount of studied and probably not really all that important in what we want to talk about. Again, this is a material that I can send you. I can email you the papers that I derive these from if you wish to have some light reading to help you sleep. But again, this shows the different types of polyphenols and their structures. And see, this is the phenol structure, and we're going to get into this, that we often just talk about phenol or phenolic foods. Phenol itself is a toxin, but phenolic compound or polyphenolic compounds are not toxic. So we want to define that. But a phenol is any 6-carbon ring right here with a hydroxyl group on it. So these are called polyphenolics because they're subunits of phenols, rings, grouped together with different hydroxyl groups or carbonyl groups hooked on. And these define, the structure determines function. So these differences in structure cause different functions. But as you see, they are very complex. You've got the flavonoids and the non-flavonoids. And this lists some of the sources that you can find these in. And as you as parents, what I would like you to hone in on is there a food in this group that your child is sensitive to? Then it could be that particular flavonoid or non-flavonoid that might be causing the problem. This might give you some clue as to what's going on with your child's digestive problems. Notice the proanthocyanidins. These are very huge. These are the largest compounds. Very hard to absorb these. So their function is going to be mostly localized in the gut. These are not really absorbed unless they're broken down into the anthocyanidins. So, and there are some bacteria that can do that. So I can provide this for you if you wish. And a lot of this stuff that you see in the tables and stuff, I can provide for you. Again, just a table I took out of a paper, and it actually doesn't even include all the different things, but it just shows you the polyphenol content by weight in milligrams per kilogram of different sources of these polyphenols. For example, this just shows that in kiwi you can find 600 to 1,000 milligrams per kilogram of caffeic acid. And it's not to say that kiwi is the only source, but it's just basically a sample. But what I want to point out is that it's very difficult to pin down the content in any one particular food because there's a great deal of variation, and this variation occurs due to the way it's grown, the conditions under which it's grown, where it's grown, how it's processed as a food can greatly affect, and that's why you see such huge ranges of these polyphenols in different foods. Fruit and beverages are our main source of polyphenols, not too surprising. Again, environmental factors can affect the polyphenol content in the food. Peeling, a lot of these polyphenols are in the outer peel of the fruit or vegetable. So if you peel them, you're going to remove a lot of that. So here, the big question and what want to focus on is there's the variability in effects seen with different diets and foods in the autism population. Why some kids are affected one day and not another. It could well be to do, maybe you peeled those potatoes one day and the next day you didn't. So you're going to get a different amount of different types of polyphenols. Maybe you served raw apples, and maybe next time it was cooked applesauce, or it was processed versus raw. All of these can have effects on polyphenols. So that's why the dietary intake can vary so greatly, and that's why some of the frustration by parents in trying to determine what's going on with their kids as far as being able to eat fruits and vegetables. So also the most common polyphenols in the diet are not necessarily the most active. So if you go back to looking at this list, you can say, oh wow, look how blueberries, wow, they're a huge source of the hydroxycinnamic acid polyphenols. So that must be what's going on with my kid. Well, not necessarily, because if we're going to touch on briefly is that depending on what type of polyphenol, it may or may not be absorbed. So it may be eaten, but it may not, it may have a low activity, it could be poorly absorbed, or it's highly metabolized by the gut bacteria, or it's just peed out or removed through the bile or feces very fast. So also the metabolites that do get into the blood and organs can be different from the native substance. This is, to me, is very important because this is where enzymes can come into play. And we're going to show that very quick. the way the body metabolizes polyphenols occurs via a common pathway, and it's actually the same pathway that the body uses to metabolize drugs. So polyphenols can actually compete for a lot of the same metabolites that drugs use, and this may or may not be a complication. A-glycones, we call, an A-glycone is a polyphenol that has had its sugar removed. So it is basically A-glycone, A meaning without, so it's without its sugar group. And it's in this form that the polyphenols are absorbed from the small intestine. However, most polyphenols are present in food in the form of esters or glycosides, which means they have a sugar group, or they're polymers like the proanthocyanidins, the huge compounds that I showed you earlier. And that's the way they come in the foods, in the raw foods. So in their present that they cannot be, or usually cannot be absorbed from the gut. So in order to be absorbed, these polyphenols have to be hydrolyzed by intestinal enzymes produced by the intestinal epithelia, or the probiotics found in the colon. And once you remove the sugar groups on these polyphenols and make them A-glycones, they can then be absorbed. And that's when and by removing those sugar groups, then they become active. So, but that's not where it stops. Because once those polyphenols are in a glycone form without their sugar, they can then be conjugated through methylation pathways, sulfation pathways, or glucuronidation in the intestinal tract or the liver. And this is a detox mechanism, and it limits the toxic effects and facilitates elimination through bile and urine. Not to say that polyphenols are toxic, but at some point they have done their job and they need to be eliminated. So this is how the body processes drugs. They methylate and sulfate or add a very complex carb group to it. And this basically tags the compound for removal. Now, as a lot of you may know, sulfation processes in kids with autism can be very low. That's why Epsom salts helps. Glutathione is dependent on sulfation. Methylation is now coming into play, where if you heard Dr. Liz Mumper's presentations, she emphasizes both methylation and sulfation pathways. And so these come into play with the polyphenols. And this is what I find so interesting, and also which makes it so complex. The overlapping between something as simple as a citrus or vegetable food, and then you get into all these other processes that you've heard about. So you can't take everything you hear, you can't get tunnel vision and concentrate on just one thing. Oh, methylation or sulfation is a problem. It's all combined. You have to look at the whole person and the whole situation, from the food you're eating to the condition of the gut, to the type of probiotics, bacteria in the gut, to the basically which enzymes are working, which are not, and the integrity of the metabolic pathways that involve the process of adding sulfate groups or methyl groups to these compounds. So that is what I find so fascinating, and is such to me, a rich area of future research. Intestinal absorption. In foods, the flavonoids, except the flavonols, are found in the glycosylated form, which, like we say, prevents them from being absorbed. Now, only the A-glycones and some glucosides can be absorbed in the small intestine. That is, they don't have their sugar groups, so they can go ahead and be absorbed early on in the small intestine. But certain polyphenols that are linked to a sugar group, such as rhamnose or arabinose or xylose, these are very complex carbs. If they're attached to a polyphenol, they are not going to be absorbed in the small intestine. In fact, they have to go to the colon, and hopefully the correct bacteria are there, and then the enzymes produced by those bacteria can then hydrolyze or take off these sugar groups, these carb groups, aramicidase, xylanase, which is an enzyme found in enzyme supplements, might be helpful in removing those. But this is the job of the probiotic in the colon. And then once those carb groups are removed from the polyphenol, they can be absorbed. But since they're down in the lower end of the GI tract, down in the colon, you don't get as much absorption of these compounds. So that's a hindrance. Now here's my point. If we do have enzymes, oral enzymes or fungal enzymes that you can take orally and that do work on these polyphenols with these sugar groups, can they then be absorbed early on and produce their good effects? Basically give their antioxidant properties earlier on. I don't know. It's a possibility. The proanthocyanidins, which were those very large compounds, again, because of their size, they're very poorly absorbed, and they may just produce their effects locally in the gut, since they are likely not absorbed systemically. This one group, hydroxycinnamic acids, they have ester groups when they are in plants. Plants basically, these are plant compounds, so the plant esterifies them or basically puts an ester group on them. And humans don't have the esterase enzymes, the correct esterase enzymes to remove that ester group. Again, it acts like a sugar group and keeps those compounds from being absorbed. Again, colonic flora or perhaps fungal enzymes might have an undocumented enzyme in the commercially available enzymes that can do the same thing. That it can act as an esterase and remove that ester group, and then the polyphenol can be absorbed. And again, I want to stress that once absorbed, the polyphenols are metabolized by the same pathways as drugs, hormones, and neurotransmitters. So they can compete with these drugs and such. And sometimes the polyphenols could even induce these pathways to work better since they are there. And that's one thing that if the compound is present in the body, then the body has to develop a way to remove it. So if that pathway is there and kind of lazy, maybe ingestion of some of these polyphenols will stimulate the pathway to work harder and better, and that may help in some of the pathways involved in this. Okay, very briefly, I want to touch on the phenol problem. Again, I showed you that the polyphenolics contain the phenol structure, but phenol itself is a toxic compound. If you ever use camphorphenique, it contains phenol, which you can put that on a cold sore or a cut, but you can't ingest it. It's not good. Phenol or phenol itself is a toxic compound. Here's an interesting point. Phenols can be produced in the gut if protein's not broken down. If it starts fermenting in the gut, you produce phenol. Now, but the gut has a way to inactivate phenol. Again, it involves the sulfation pathways, and those are found in the mucus lining of the gut. Again, so these enzymes, these phenol sulfotransferase, are found in the gut mucosa, and these are what sulfate phenols, and once they're sulfated, they can then be eliminated. It makes them water soluble, and they can be eliminated by the body. Interestingly, the phenols and salicylates are very similar in structure. So to me, this says that you need to make sure that we're breaking down protein, that it's not fermenting in the gut. This may be one of the reasons why proteolytic enzymes supplementation seems to help. You're just reducing some of these toxic areas, especially if there's gut damage. If the gut mucosa is been compromised or damaged, if there's protein fermentation, then you're going to get some toxicity. So again, here's another area that enzymes maybe help. Okay, and again, I apologize if this seemed to be scattered and all over the place. But again, let's just go over some things. On the lectins, they're very abundant in food. They can bind to gut cells and cause it to be leaky. They also are involved in IgE and IgG reactions as well. Oxalates depend on gut flora for removal and degradation. So that's basically the bottom line for oxalates. So again, one reason why probiotics can be very helpful, and potentially this might be an area where oral enzyme supplementation can be helpful as well. Phenols are toxic, but the polyphenols are nutritional, so we want to emphasize the difference. These are different compounds. And really when you break down polyphenolics, you don't break them down to phenol. So that's one thing you don't have to worry about. You do require a healthy mucus layer in the gut for the sulfation enzymes to work to remove the phenol. And polyphenolics, as you've seen, are a broad category of compounds. Most are involved in antioxidant reactions. But for them to work well, they have to be absorbed, and to get them absorbed requires removing their carbohydrate groups in order for them to become active. If the gut is damaged, those particular enzymes needed to modify the polyphenols may not be present. If gut damage there, or if gut flora is missing, or if yeast and Clostridia is a problem, then you may need extra help, and perhaps enzyme supplements can be helpful in that area. And again, we distribute enzyme products, and I formulate enzyme products, and I would like to be able to tell you for sure that taking oral enzymes will be the answer to your problems, but I'm not that confident. I have to see it in, I think the anecdotal evidence is very promising, but we do need to confirm this by some good studies. So I'm hoping that we can get some good research on this, and at that, I think I will shut up and let others. Cindy can ask me some questions that you've given, and I think I'm going to, oh, wait, I'm sorry. There's a contact information. I put a little thing here. I'll be happy to email you the articles I've used in researching for this presentation, and the other info I have, including labels if you want to know which foods are high in polyphenols or which are such. But do bear in mind, I know it's very easy to go out and get on the internet and repeat what you've heard someone say, but just bear in mind that this is a work in progress. A lot of this is based on my interpretation of data from current literature. So as that data changes, as more research comes out, I do reserve the right to change my mind on how we interpret the data. So I just basically want to emphasize that there are a lot of people interested in researching on this. Certainly I think we're going to give it more attention than we have in the past. We've been more emphasis on DPP4 and gluten and casein problems and that thing. But I think the phenolic area, or the polyphenolic area, I think we should distinguish that, differentiate a little bit better. I think is another area that's going to come into play, because I think it dovetails in with the oxidative stress problems a lot of these kids have, as well as the sulfation and methylation pathways as well. So with that, Cindy, do you have questions you want to give?
We do, and I know that we have gone over the time that we were going to try to stick with. So those of you who have to leave, you can email us your questions, and we hope to post this recording on our website in the near future, so you might look there also. I have many questions. How about the first one? What do phenols do to the brain receptors?
Okay, again, we want to differentiate between phenols and polyphenols. From what I've read, there's little evidence to suggest that polyphenolic compounds, even absorbed, can cross the blood-brain barrier. So I don't think they're going to have a direct effect on the brain itself. Now they can have an indirect effect by limiting oxidative stress, keeping compounds that could damage cell membranes from occurring. From the phenol issue, if the phenol or the toxic phenol is not sulfated and addressed, then that could possibly, that being a very small compound, could possibly cross the blood-brain barrier. So phenol itself might play. I'll have to look into that a little bit more. That it might have an effect on the brain. But polyphenols, no, I don't think they're directly affect. They're just going to keep oxidative stress to a minimum if they are present in good amounts.
Okay. Is it necessary to use lectin lockers if you use enzymes?
Someone asked me that question. There was a product called Lectin Lock they were asking me about, and I basically just wasn't that impressed with the product. But then that was before I started looking into that, and I don't know. I saw the ingredients on the product, and basically it contains things like okra and mucin and mannose. So if the particular lectin is a mannose-binding lectin, then ingesting higher amounts of those compounds might, again, bind up the lectins, keep them from being harmful. But there again, you're just targeting one small type of lectins. Again, the lectins are a very diverse group. I don't think we can get them all. I think the Lectin Lock product wouldn't be harmful. I don't see any downside of taking it, except it might hurt your wallet, but it might be something you want to try. The enzymes aren't going to affect that one way or another. Again, I think the enzymes probably provide of these groups that we've talked about, probably provide the least help for lectins, except that if you're breaking down the foods you eat, you're going to produce same sugars from the carbs that will act like lectin lock, basically providing a variety of sugars that can then bind to the lectins and keep them from performing any mischief. So again, I'm not sure, but again, it's one of the things you can always try.
What is the connection, if any, with autoimmune disorders?
Ooh, yeah, we didn't even touch upon that. It's very high. The lectins, as well as the phenols, and I'm busily looking through one of these papers, but yeah, they can bind to certain areas. Let me see right here. Yeah, in this one paper in British Medical Journal, I'll just read verbatim. Of particular interest is the implication for autoimmune diseases. Lectins stimulate class II HLA antigens on cells that do not normally display them, such as pancreatic islet and thyroid cells. The islet cell determinant to which cytotoxic autoantibodies bind in insulin-dependent diabetes mellitus is a disaccharide N-acetyl lactosamine, which must bind tomato lectin if present, and probably also the lectins of wheat, potato, and peanuts. Certain foods such as wheat and soy are indeed diabetogenic in genetically susceptible mice. Insulin-dependent diabetes, therefore, is another potential lectin disease and could possibly be prevented by prophylactic oligosaccharides. Another suspect lectin disease is rheumatoid arthritis. The normal human IgG molecule possesses carbohydrate side chains which terminate with galactose. In rheumatoid arthritis, much of the galactose is missing so that the subterminal sugar, N-acetylglucosamine, is exposed instead. So basically what he's saying is that yes, there is evidence that lectins could be implicated in certain autoimmune diseases, basically going through the stimulating the HLA antigens on cells that normally would not be stimulated. So there are other papers talking about mannose-binding lectins that increase the risk of, actually in the deficiency of mannose-binding lectin can increase the risk of disease-specific complications. So again, it just depends. We're going to paint in broad brush. In some areas, lectins can be helpful. In others, they may not. Again, it just depends a lot of genotype, environment, general health of the gut. So again, yeah, but yes, there can be a connection between autoimmune problems and lectins, and probably the polyphenols as well.
Can you mention again which enzyme formulation for oxalates?
Well, again, the standardized enzymes that are available for commercial production don't address the particular enzymes for oxalate degradation. But 10 years ago, we didn't know that there was a particular peptidase enzyme with a DPP4 that addressed glutamorphin and casomorphin. We didn't know that was already present in some of the protease enzyme blends. So I'm guessing here, but I'm thinking some of the xylanase and cellulase enzyme blends out there may contain, and I emphasize the word may contain undocumented enzymes that could be helpful in oxalates degradation. But right now I'm skeptical. But what I do want to emphasize is that there is a bacteria in the gut, the oxalobacter bacteria, that can produce the enzymes to break down oxalate. So the important information here is get the gut healthy. And enzymes can be help in getting the gut environment healthy so that probiotic and bacteria colonies the good ones can proliferate. So you can keep out the competing yeast and the bad bacteria like Clostridia and Pseudomonas, and that will allow the good bacteria you need to break down the oxalic acid to thrive better.
Do many of the non-gluten starches contain lectins?
I think so. I think from my reading, potatoes are large or have a high amount of lectins, as do tomatoes. But just about all plants, just about all vegetables and many fruits do contain some amount of lectins. Now, some of these lectins some are destroyed by cooking. Others are modified by fermentation. Some of the like soy, tofu, which is fermented soybeans, are going to be much lower in lectins than raw soybeans, or the edamame, which is basically just steamed or lightly cooked. So, and just from personal experience, I love sushi and I love the edamame. I always like to order it before, but seriously, I've got to not pig out on the edamame, or it seems to affect my digestion. But I seem to counter that if I can take a lot of or Enzyme Prime, which has a lot of amylase, and to counter the inhibitors in the beans. Even soybean has enzyme inhibitors as well that are heat stable. So basically, you can counteract the negatives of certain foods, whether they're lectins or enzyme inhibitors, by taking in extra enzymes.
Regarding the mucus layer in the gut, is there anything that helps replenish beneficial mucus, similar to taking probiotics to replenish good gut bacteria?
I think there are. I would have to look into that, though. I'll write that down, and if you can send me an email on that if you wish, and I'll share whatever I find. I think there are some things that are mucogenic, basically stimulate mucus production. The goblet cells in the GI tract are what produce mucus, and they are there on the forefront, on the inside layer of cells, right next to so they basically are in contact with what comes through the gut. So if they are damaged through yeast or inflammation or such, then the amount of mucus may be lower. So again, it's basically controlling inflammation, getting rid of the yeast and those things. And enzymes can sometimes, or can often play a role in just the general housekeeping of the gut.
What about soaking and sprouting beans and legumes?
Yeah, that the soaking can remove a lot of the lectins and toxins in plants, and I think sprouting can do that too. And I think that's why they're healthier than the nuts themselves, the seeds themselves. So I think that is true. I haven't run across it recently, but I think in the back of my mind I've run across it in the past. So yeah, but again, I want to say I know there's a big segment out there that really are pushing raw food diets, and I think for some that's good, but on others you just have to understand what you're doing when you're eating raw foods. You're going to be taking in a lot of these compounds. You're going to get a higher dose of them. If they don't bother you, then it's not a problem. But if they do, then that may be. And I know there's certain raw foods that I cannot tolerate. So in those instances, cooking is fine.
Are you familiar with a test that can point to reactions to polyphenols?
Well, no, I'm not sure there is any really good tests other than maybe some allergy testing. Again, I think what I want to convey here is maybe to help the parent be able to discern, is it a lectin problem? Is it oxalates? Is it polyphenols? Or, is it a pure phenol problem from protein fermentation? Again, you just have to do some footwork looking at patterns of reactions, which foods. Interesting, if you can, look at the form of the food. Does a raw apple cause your child to react? But if they eat applesauce, they don't. That may tell you something right there. So I don't think there's any formal testing one can do, at least routinely. There may be some companies or doctors out there on the edge that may have something that, while not universally accepted, may be something that they swear works. I don't know. I'm not sure.
If you are not sure your child has a phenol issue, would there be any harm in trying No-Phenol or try Enzyme?
No, there shouldn't be any harm at all. In fact, that's what we espouse. The nice thing about enzymes is you can always try it, and because there's no downside. Again, they are the absolute safest supplements on the market, because they if the thing they work on is not there, they just don't do anything. They just become paralyzed and just go on and are treated as a food themselves. So yeah, I mean, there's no harm in trying.
We have a lot of questions. Here's another one. Can you check for the enzymes present like you can for bacteria?
Enzymes in the gut, it's very difficult. Some doctors do, they test the feces for certain pancreatic enzymes, and but that's not always advantageous, because different pancreatic enzymes have different lifespans. And sometimes, what comes out as feces, the enzyme may be perfectly active a day, 20 hours earlier, but by the time it comes out as stool, it may not be present. So other than an invasive procedure where you can monitor pancreatic enzyme output, basically putting a tube down the child and into the small intestine and giving a shot of secretin, which stimulates pancreatic enzyme production. So it's not an easy thing to do. So it's oftentimes you just have to assume, or just if you question whether a certain food is tolerated or a certain group of foods. You know, if proteins are tolerated, it's probably has certain, the proteases are probably fine. If it has trouble with carbs, then maybe the carbohydrates. And usually, in general, the enzymes that are most likely to be missing or deficient are the carbohydrates type enzymes, so the enzymes that are produced by intestinal cells, especially if there's gut damage. So things like lactase or the peptidase, the cells that produce these enzymes in the intestinal tract may be the ones that are deficient. The pancreatic enzymes, I mean, all things being equal, the pancreas puts out more than enough enzymes to take care of digestion in the gut. It's just that what we espouse is where digestion occurs. For gluten and casein problems, we say you have to address gluten and casein while it's still in the stomach. So that's prior to where the pancreatic enzymes come into play. But things like lactose intolerance, that's where lactase enzyme, which is produced by the intestinal cells, not the pancreas, is low. And so sometimes it's a program, your body just programs to stop producing certain enzymes, or if you have gut damage, the cells that produce the enzyme, they're not there because of inflammation or gut damage. And then that could equal an enzyme deficiency.
My son is affected when eating bananas, very hyper and stimming. Is that from high phenol levels or high sugar level in the bananas?
That's a good question because you have high amounts of both. Again, you you just kind of got to be a detective on these. You might try cooking the bananas if you can, if there's some way. If he tolerates cooked bananas, then that might tell you that, you know, maybe it's the phenols. Or if he can eat a very ripe banana, which would be very high in sugar, but the phenols or the polyphenols may be lower in the very ripe banana versus a green type banana. So those kind of things is what you look for. If you can change the processing of the food, then that might give you some indication what the problem is.
How about one more question?
Okay.
Would taking enzymes to counteract the effects of the lectins change the allergic reaction that lectins can create?
Totally unknown. Sorry for that to be the last question.
I can give you another one if you want.
Well, I mean, I hate to end on an I don't know answer, but the lectins are very resistant to proteases. Protease enzymes are probably not going to do much to break down lectins. But the thing is, since lectins are selective for carbohydrate groups, if you can change or damage or cleave off the part of the lectin that interacts with the carb group, whether it's on the lectin or whether it's on the food, or if you can supply something that will block the lectin from it. And again, the carbohydrates enzymes, for example, amylase, we have a lot of starch. Usually every meal we have a lot of starch. So an amylase enzyme is going to work very quickly, very well to break down the starch into the oligosaccharides and the sugars that would bind to and just block lectins from interacting with anything else, parts of the body, part of the gut wall, receptors or enzymes. So again, theoretically, that's why they would. But is there a specific enzyme to address lectins? No, I would never say that.
Okay, you know what? I want to give you one more question because I think this is kind of intriguing. If a child has no lactobacillus in the stool, might you assume that there is an oxalate issue?
Not necessarily stool. I'm sorry.
That's fine. Go ahead.
Well, again, it's sort of the same way as enzyme testing in stools. most of the fecal matter is dead bacteria, and a lot of that is dead probiotic bacteria. So sometimes stool analysis is, you have to take it with a grain of salt, which that doesn't sound right to say, but there's, it's waste material, and I'm not sure if looking for live bacteria and live enzymes is the way to go. Again, that's the problem with probiotics, is that certain strains work better in different people. It's a very individualized thing. That's why there's so many different probiotic products out there that all seem to be good. It just depends on the type of gut you have, whether it's like building birdhouses to attract a particular species of bird. If you want purple martins are very, very picky about where they live. So some probiotics are very picky about which guts they're going to thrive in. So same thing, and that's why sometimes it's very hard to tell, except the things, your bowel movements.
Hello, I lost you. Can you hear me? Well, we're going to go ahead and wrap it up there. I apologize if I'm speaking over Dr. Houston. I've lost the audio for him. So if you all can hear me, thank you for joining us. And if the recording went well, we will post this on the website in the near future. You can look under What's New on the home page. And you're welcome to email me or Dr. Houston for the slides. And I may possibly post those in the articles section, so you might check there in a few days too. Thanks, and check back in the future for webinars on other topics. Have a nice day.